So my name's Keith Uloth, and I'm going to talk about the BC Pests Pest Monitoring Project first, and just kind of give an outline of what we do up here in the BC Pests, and then kind of go into some of the work that I've been doing with Karen Tabe over the past 2 years down in the Vanderhoof area. So, let's see. Seems to be frozen. There we go. So I would just like, before I begin, to thank my funding partners, our funding partners for the project. So the Peace Region Forage Seed Association, the BC grain producers, both the provincial and the federal government, and then just the funding association. So Investment Agriculture Foundation of BC, Climate and Agriculture Initiative of BC. They've been big partners in kind of keeping going this project up here in Peace. So project objectives for the project when it was initially founded were just to kind of monitor the pest populations in field crops over a given season. We have quite a diverse amount of field crops up here, and so the focus was to try and start with just annual cropping and carry on from there. Second objective was to obtain data for populations of insects and diseases for field crops in the region, and also to provide support to researchers in conducting research of insects and diseases in the region. And then finally, to develop some baseline data for new and emerging pests within the region. So a little history of the project and how it got started. In 2014, with funding from BC Agriculture and Food, Climate Action Initiative, the Peace Region, the Peace River Regional District, the BC Grain Producers, and the Peace Region Forage Seed Association. They put the project, there was some funding provided. It was headed by the BC Grain Producers for some funding, and it was with the help from Agriculture and Agri-Food Canada, Beaver Lodge. There was a bit of a void in the Peace region on the BC side, with cutbacks due to changing of governments, stuff from my understanding. There was a lack of monitoring in the region, and so Jennifer Otani and her integrated pest management lab helped the grain producers basically start this project off the ground and grow it as it has over the past few years. And to start, it primarily consisted of monitoring annual crops, which I had previously mentioned, mostly in canola and wheat. And then in 2016, that round of funding ended from the Climate Action Initiative. And so new funding was obtained for the project through Investment Agriculture Foundation, and the BC Grain Producers and the Forage Seed Association continued to fund the project through their levy dollars. It changed hands as the administrator did, with the Forage Seed Association taking over control. The hope was to continue to monitoring annual crops and eventually transition into monitoring some forage as well. I started at the fall, in the fall of 2016, so I came on just as the season had ended. So I didn't actually get to do any monitoring, which was a little disappointing for me, but I did get to my feet wet with a little bit of grasshopper surveying. So it was nice to do and a bit of a wake-up call for me up there in the Peace region. And then in 2017, we started to monitor a little more forage crops grown for seed, mostly sticking with creeping red fescue and some red clover that was being done in there. And then by the end of our by the end of our funding in 2018, we had grown the project to 6 canola fields, 6 wheat, 5 fescue, and we had started to do some disease surveying mostly in the forages, but we were starting to do some in field peas, which was starting to creep into people's crop rotations, and some of the other cereals as well, and some cereals as well, sorry. And then on to 2019, we actually didn't receive a lot of funding through the CAP program. So we kind of had to backpedal, and fortunately enough, we had a bit of a saving grace through our provincial government. Tracy Heppelhauser came in and helped us a bit. So she gave us some funding through the Biosecurity Fund, which allowed us to maintain some monitoring within the region. So we were currently, we had just started doing five of each. So we did 5 canola, 5 wheat, and then 5 creeping red fescue fields to try and keep some of that data flowing for ourselves and for researchers as well. And then in 2020, we got a bit of an insurgence of money into the program, where we secured some money from Climate and Agriculture Initiative. And we were able to scale up our monitoring to include more forage seed crops, more annual crop types, and increase some disease survey stuff that we were wanting to do. And this was we were allowed to, well, not allowed, but we could hire a student. So most of the project is done through one person. So myself, I usually do most of the monitoring. And then with the increase of a student, it was nice. We were able to upscale a bit. And we also added some horticulture monitoring. So we partnered with the Northern Cohort up here out of Fort St. John. And we're doing some monitoring of market gardens, berries and apple orchards, just to try and develop some baseline understanding of what insects we might be finding in those crops that we weren't already finding in some of the field crops that we had been looking at before. So what our monitoring kind of consists of, we do a fair bit of passive monitoring with traps. So we do use a fair amount of different styles. So Delta, Jackson traps, those yellow sticky traps that you see there on the bottom right. We use those quite a bit for most of our trapping. And then we use also some pitfall trapping or ground traps for for click beetle populations, and also looking at carabids and staphylinids that we have in the area, so beneficials. And then we also have some unit traps set out that we use. We tend to use a pheromone with those ones, and also with some of our Delta traps as well. And then with our sites, or with our traps that we set out, we add our trap lines, as we like to call them. We have some active monitoring, so we do active scouting, so looking for insects that we don't necessarily trap for, but still may be causing damage within the field crops. We also look at diseases as well, and with that we do sweep netting too, and we also take pictures of the damage and any other insects that we may see possibly out in the field that we couldn't catch in a sweep net. So this is a pretty big list of insects that we look for, but we do this mostly through some of our traps, and also some of our sweep netting that I process over the off season. So in field peas, we're currently looking for pea leaf weevil. You can see that we're also looking at that one in our forages as well. So that weevil tends to be found in some legumes in alsike and alfalfa and red clover. And it's currently not found in the BC Peace region, but we actively monitor for it. Wheat midge is a big one up in the Peace region that we look for. In wheat, you can see that canola, there's quite a bit. It's a pretty high price commodity, so the BC grain producers have us kind of looking for a lot, and a lot of that data that we collect also goes into the Prairie Pest Monitoring Network, which is a Western Canadian project, and that's through the federal government. You can see that we also have some cross commodity pests, so grasshoppers, lygus bugs, aphids are ones that we look at too. And then we do a bit of Japanese beetle monitoring. That's an insect that's not currently found in the region, and hopefully not. But we do that one through Beaver Lodge. The federal government asked us to do some traps along major highway corridors for them, so we do that. And then our forage pests, you can see our forage seed. We look at red clover case bearer and red clover. We also look for other case bearers. Pea leaf weevil, European skipper is one that we've seen over the past few years expand into the region, and so we actively monitor for that one, and provide some research data to Beaver Lodge as well for that. And then the other one is silver top. And that's actually a symptom of what we find. Jennifer Otani and her lab up in Beaver Lodge are trying to find the actual insect pest that might be causing it. And then you can see all the beneficials that we look for, and we don't actively trap for them. They're kind of either involved in some of the bycatch that we do. That happens with all the trapping that we do, and also just kind of with some of the active monitoring that we do within the field. And then the horticulture is new to 2020. And I have to say thank you to Tracy and Susanna for helping me with this one, because I probably would have been lost to start with. And so they helped me kind of guide through in trying to figure out some of the trapping stuff that we would do within this horticulture project up in the Peace region. So we started with some spotted winged Drosophila. We've been doing it for 2 years now and haven't actually found any suspect flies. But that is one that has been found, I think, in the central Alberta, in the southern Alberta area, and seems to be spreading through the prairies as well, in other parts of the prairies. And then codling moth, we had some suspect damage in 2020, so we put pheromones out last year, but we didn't actually get anything, which is a good thing. And then just with the market garden, looking at a variety of insects, and we try and pick an insect every year to see if it is in the region or not. And Tracy and Susanna usually give me some help with that. So the other part of this project is diseases. And that also goes with some of the site monitoring of field crops that we do. So we do also do disease surveys in the field crops through the region. So we'll, for example, with the club root surveying that we see there, that we did in 2018 and 2020, this year we were able to, with some funding, sorry, excuse me, I have a bit of a cold. We were able to secure some money to do some soil sampling for club root. It's not a disease that's found in the region, but it is one that is, if it does get into the region, it can affect canola pretty heavily and affect a producer's rotation and everything. So it is a pretty serious disease. So we were able to do some soil sampling. We soil sampled about 64 fields this year, and usually on average before that we were only doing about 20 or so fields, and it wasn't soil sampling, it was mostly plant inspection. So you can see that we also do field piece forages. The forage disease sampling we do on both sides of the piece. We do in the Alberta side as well, and that's kind of working in conjunction with Beaver Lodge. They have a pathologist at their lab there that specializes in forages, so we go out and we gather some stuff for him, and we also send some samples off now to UBC. So there's a researcher down at UBC that's interested in some forages and also cereals diseases as well. So you can see again at the bottom of the slide there, we send our samples off to Beaver Lodge, which is Agriculture and Agri-Foods Canada, and the Provincial Plant Health Lab, and then some researchers with specific interest in certain diseases. So just to kind of give people who haven't been to the Peace region an idea of how vast it is, you can see, and I'm not sure if you can see my mouse or not, but you can see Chetwynd there on the bottom left. You can see Dawson Creek kind of on the bottom right. I have a bit of an orange arrow there to give you a guide. Fort St. John is that little blue arrow, and that's kind of the area that we monitor on a weekly basis. So we have about, I think we have around roughly 15 sites per day, and we monitor through the week. We're checking traps constantly. So you can see what we do an average travel during the week of 900 kilometers. And we travel as far north as up to that Presbyteau area, which is about 160 kilometers north of Fort St. John. And you can see that the Peace region, and this is a Wikipedia, so I don't know how accurate it is, but it says it's about 2.5 million acres of agriculture and production. And we do a bit of monitoring in also pasture and hayland as well, in both the north and the south Peace. So the other half of this project that we've started to really focus on since 2018 was our communication. So in 2016, when I started, the current form of communication to let our producers in the area know of updates was an email about once a month throughout the season, and that was kind of just giving them an idea of what was being seen in the traps and what they might be of concern with. And so in 2018, when we were able to hire a student, we had a bit of a shift. At that time, we had a coordinator come in as well, who was amazing, and she thought of an idea of using an SMS update, so a text message, to contact local producers and just give them a bit of an idea and a more accurate, I guess, and up-to-date idea of what was going on in the field. And we also created a Facebook page and also a Pest of the Week, which was published in a local paper on a bi-weekly basis. So it was amazing. We had lots of positive feedback. And then in 2019, with not acquiring some of the funding that we were hoping for, we kind of had to scale it back a bit. So we went down to the SMS updates. We kept those going. And we also started to send those updates out through the BC ministry. So Laurie Vickers kind of came to me and said, well, we can send them out this way as well. And I thought, well, that's amazing. So, because we want to reach as many people as humanly possible. So we did that. And it seems to be a pretty good success. And so in 2020, with hiring a student again, we brought that all back and we continue to use that. So we currently have about 45 users that receive SMS updates on a weekly basis during the growing season. We have about 71 followers on our Facebook page. We're always looking for more if anybody's interested. And so, yeah, so the SMS update here's an example of it. Sorry, I just had to drink some water. So we have the time period you can see there, and this one's from this season. So from June 28th to July 4th, and it highlights what we've seen in the monitoring, in our traps and in our sweeps and out in the field, and just kind of also gives an update with what other people might be seeing. So the Prairie Pest Monitoring Network is one that we also watch closely with, and that kind of, you can see that at the time, I think we were in the heat bubble was starting. So Lygus bug warning from the Prairie Pest Monitoring Network across the prairies. And we were also starting to see some of that trickle down into our region as well. We also do provide growth staging of canola, wheat, and peas using a BBCH scale for agrologists. And we also do growing degree days using our local weather station. So we have a BC Peace Agri Weather Network, and that's 21 stations that have been set up through the Peace region to try and get some more localized weather. So we use those in conjunction to try and determine some pest issues. That website has some tools also for producers in the region that they can access for IPM. And we also provide some links to websites and useful information for integrated pest management, monitoring or scouting protocols, and some tools. And those get sent out with our email update that we send to Brenna Shields or Laurie Vickers. And then the main body will be what is sent through the text message. So some of the research connections of the project, of course, the big one for us is the Prairie Pest Monitoring Network. A lot of what we do feeds into that large scale monitoring. And then on some smaller scale stuff, we have just finished, I think we're in our last year with the Peace Region La Terre Day survey. So that's with Wim van Herk out of Agassiz. We've been doing some, I think since 2018, some work with him on trying to determine what click beetle species are found in this region. And then a lot of what we do is tied through the Beaver Lodge Lab, IPM Lab. We partner with them quite a bit on stuff. So we've been doing some silvertop research in creeping red fescue, some stem eye spot disease stuff in creeping red fescue as well. Our club root survey, as well. And then stripe rust in wheat and other cereals. That kind of goes with the cereal and forage diseases through UBC. That's with Professor Gucharn Brar. He's been doing some work, I think some genetic work with some of those diseases, and then the establishment of presence or absence of horticultural pests with the northern cohort, just to name a few of the stuff that we do. So some of the challenges for the BPM, the big one for us is funding. We are always trying to acquire funding. We seem to be get on a 2-year rotation for some of our funding, and it's always a bit of a challenge to try and get some long-term funding for the project. With that said, though, when we do, it's great. But with the Peace region being so big, I think it takes up about 1/3 of the province. We have trouble covering the region with the number of sites and surveys. So it's and always the changing of demand. So there's new crops that are being introduced every year. I think the latest ones are faba beans I've seen growing up here last year. And there are some guys trying to grow a few other crops that have been mostly stuck in the Saskatchewan and the southern areas. So it's always nice to see, but it also means that we have to try and expand our monitoring for some pests that are more specific to them as well. And then the other one is knowledge transfer. So we are always trying to create tools or raise awareness of insects in the area for farmers, help with some identification and information as well. And then just those seasonal challenges that everyone faces. So the weather and the ever-changing pest pressure and disease pressure from weather or from how they migrate in through the season. So I'll move into some of the work that I've done with Karen Tabe in the Vanderhoof area. So in the winter of 2019, 2020, Lori Vickers kind of put me in touch with Karen Tabe. Karen had said that there wasn't any active monitoring going on in the region, and she was hoping that I would at least be able to come and talk to some people, some local producers in that area, and kind of give a comparison of insects that we may have in the Peace versus the Vanderhoof area. So I came down in June and gave a talk and just kind of got an idea of what producers were seeing out in the field. And then in July, I came back and did a bit of a field day. We visited some fields around the area. The big takeaway from that one is that we identified that European skipper was in the region. There were some farmers that had some hay crop with some Timothy in it, and they were noticing that these orange butterflies were flying around. So we identified that that was European skipper in the area. It actually came out in the Peace region around that same time that people were starting to notice it as well. So it was a bit timely for me, which was nice. And then in 2021, I did again 2 visits, one in May. We put out some yellow sticky traps and also some pitfalls at sites around, just to try and get an idea if we were going to see any pests or any insects that might be of interest. We also noted at that time that we were seeing some grasshopper or some early instars out in the field. So I had taken some samples to identify. And then when I wanted to try to confirm my ID, I contacted Dan Johnson, an entomologist out of Lethbridge, who said that it would probably be a lot easier to do the adults. So I came back in August and we did some grasshopper sampling at some sites around the region and did ID on the adults. And I'll have my results for you guys in just a second. So in this coming year, I have 2 scheduled visits with Karen as well to try and continue that grasshopper ID work, and also to look at some other forage pests that might be occurring in and around the region. And so I guess I'm going to have to kind of coordinate with Dezene as well to maybe see if there's some interest there as well. So, so the sticky card findings and the pitfall findings from May were really interesting. The sticky cards kind of were a little disappointing in the sense that all we really found were flies. We got a lot of March flies and crane flies and mosquitoes. There was some thrips, but we did find some nice rove beetles, which are great predators of aphids and caterpillars, maggots and mites and insect eggs. And then I also found some parasitic wasps, which were cool. I'm hoping that I can send some of those samples off to someone to ID a little further so that we can get some species identification on them. But they're also great predators of aphids and caterpillars and maggots as well. So it was nice to see that there was a good healthy population of beneficials in the area. And then in the pitfalls, we did find quite a few click beetles. The species that I was able to identify pretty easily was Hypnoides bicolor. It's a pest that we have up in the Peace region of our small grains. So wheat, it's also a pest of potatoes and raspberries and most things that have a root system, and grasses. But the interesting one was that I found a Stenicera species, and I'm hoping to hear back from Wim van Herk about further identifying that down to a species. And then some of the bycatch that we found in there, that I found in there, was beneficial. So I found a good healthy population of ground beetles. And they're predators of slugs, caterpillars. They like eating other insect eggs and snails, to name a few. And some of the genuses that I found were Pterosticus. I did find some agonums as well, and a Carabus species as well. So, and then again, I found some more rove beetles, which were great. And I have some pictures of parasitic wasp there on the bottom right. And then that one on the top right is a rove beetle that I found up in the BC Peace. It's quite big. The ones that I had found on the sticky traps and in the pitfalls were a little smaller, but still cool nonetheless. So the other part of what I was doing in the region, in the Vanderhoof area, was the grasshopper stuff. So in 2020, it was when I did the presentation on the grasshoppers that we had up here in the BC Peace. It was identified that you had both the Melanopolus bruneri and Melanopolus sanguinepes. So the migratory and the Brunner spur-throated producers in the Vanderhoof area said that they had both. And so I was trying to figure out if they had both, or maybe it was just some their populations were a little higher of one than the other. So when I came back in 2021, we did a bit of a small scale survey and collection, and the grasshopper numbers were quite high in 2021 in the Vanderhoof area. And so the predominant species that I had ID'd in that area was the Brunner spur-throated. So the hope for this year in 2022 is to come back and see what species are now prevalent in the Vanderhoof area in this summer, because Brunner spur-throated, and so you can see that I have a little bit of a chart. I'm sorry if it's a little confusing, but Brunner spur-throated are biennial, so they have a life cycle of every 2 years, whereas migratory species are occurring every year. And one grasshopper that we did find in the survey that we did last season was a migratory, so they could both be in the region. It's just would be interesting to know the actual numbers, I guess, or how the populations are between the two. Some other differences between them, and these are literature related differences, is that migratory is supposed to be a little more diverse in its ecosystem. So they tend to spread out a little more throughout the environment, whereas our Brunner spur-throated is a little more picky. They tend to like the sun, a little more dry on top of hilltops, little more open area, but both will both feed on, they both have a pretty general diet for the most part. And those bottom pictures that you see there are of the grasshopper's abdomen. That's just the circus or the cercis. That's what we use to identify them. The species are very similar looking when you look at them just in a general sense. For males, you have to look at that little cercis disc and compare the differences between the two. And in talking to Dan Johnson, there are little tips that you can use to identify the difference between the males, or sorry, between the females as well. But that's kind of, some people use dissection to tell the difference between the females. So that's kind of what I found in the 4 visits that I've had in the region. And I'm hoping to have a few more in the next couple of years to try and help with some of that stuff that producers in the Vanderhoof area are having with issues in regards to pests. These are some tools and guides that we have that are out and available for people. The guide to pest wireworms in the Canadian prairie, that's one that's new out this year. And the field crop and forage pests guide is also available. That's a good one for lots of growers in the region and in the area. And then the Prairie Pest Monitoring Network, I have that up there for people. There's lots of good tools on there for monitoring protocols and scouting tips and all kinds of things, which might be useful to some people. So questions? I'd just like to give a special thanks to all those people involved in the project and supporters of the project.
Perfect. Okay. Yeah, thank you very much, Keith, for your talk, and Aija, I'm looking forward to hearing yours as well too. It's really great to hear about all of this monitoring that is going on and will be going on in this region. One of the things that that struck me when I first took this job, actually, quite a few years ago now at UNBC, was how fairly neglected everything sort of north of Kamloops can be in this province sometimes in terms of surveying and knowing what's actually there. And over the last few years in my lab, we've been, doing our small part to try to rectify that in a few contexts, including pollinators, which we hear from with Aija shortly, and aquatic insects as well. And this project itself, you can see a lot of our partners down below: Investment Agriculture Foundation, Canadian Agriculture Partnership, of course, the province of BC, and the federal government as well. And the funding for the grad student, Megan, who will be doing this work, comes from MyTax and the BC Forage Council in particular, in terms of stipends and travel subsidies and other things. And it's being run out of UNBC and Vancouver Island University. Dr. Jasmine Janes at Vancouver Island University is partnering on this as well. We are, what I put in the very top title there is, this is the MyTax title that we use, and it's sort of jargony, but I'm going to read it anyhow and try to explain it as I go. Enhancing ecosystem service provisioning by natural enemies of arthropod herbivores in the ecosystems of BC's northern central interior. So ecosystem services is a sort of, I don't know, a jargony way of saying the things that organisms do for us in nature. So sometimes we might think of things like flood control with trees, or slope stability, for instance, sometimes noise abatement, pollinators, of course, seed dispersal. And one of the big ecosystem services that we care about in agricultural settings, of course, is control of herbivores. And of course, there are a lot of other arthropods, so things like insects and spiders and a few other creatures as well, that act both as herbivores, eating crops, either the leaves or the fruits or the roots. And there are arthropods as well that act to eat those herbivores, so those things that are eating the plant parts. So I put a little too long, didn't read down below there. In general, we are looking to do here in the next couple of years is to identify agricultural pests and the natural enemies that control them. So predators and parasitoids that control the natural enemies in our region. And we want to find ways to encourage healthier populations of the predators and parasitoids. So the first thing we need to do is figure out what's actually there. It's hard to know how to work with these creatures if we don't actually know what's there. And Keith has shown us a really good example of some fantastic monitoring that's been going on. That's beginning to uncover that in the piece, and also a little bit further south here. And we're hoping that we can extend that as well, and also look at, of course, predators and parasitoids that way. And once we know what's there, then we might be able to find ways to make the populations of predators and parasitoids more healthy and robust so that they can provide better ecosystem services for us. So in the case of my talk, this work hasn't begun yet. It's going to begin this May when Megan starts at UNBC, and we get going with the next growing season here. So what I'm talking about here are the things that we are planning on doing. And so Serena is going to be carrying on what I would say is an ongoing umbrella activity, that being stakeholder engagement, and of course that's partly what's going on here tonight too. We really need to know what producers are seeing in their region. And I like that question earlier about the skippers, because that's a really handy thing to know about what people are dealing with that way. This part of this project took off because of people discussing grasshoppers as being an issue throughout the region over the last few years as well. So we really want to be able to talk to producers before and during, and I mean, we're going to be in people's fields and after as well. And part of that is finding fields to work in. Aija is going to be discussing some of the pollinator fields she's been working in. We're hoping to piggyback a bit on that work and maybe add a few more fields as well. So being able to have these discussions is going to be very helpful for us in terms of developing information and data and analysis. And then also down the line, I always feel that science isn't very useful if it just stays in what we would call a silo, which is different from the silos that you'd find on a farm. But similar idea. We don't want everything just to stay in there. We want to take what's in there out to use and to pass along to other people. So that's what we are hoping to do over the long term with this, and to develop a better practice that way in general. And Serena, as I show on the bottom left, is going to be leading up that portion of this work. Megan, so on the bottom left, I always show who's going to be leading up whichever portion of the work. Megan is going to be doing a couple of activities. The first activity, and it's similar to what Keith was discussing there, is to determine which arthropod, herbivore and predator species are present in the region. So which insects and spiders and other creatures are present, either in the case of insects feeding on the crops, or in the case of predators and parasitoids, things like spiders and small wasps and some of the beetles that Keith was talking about as well. What is actually present in the region? And by the region, we are defining it as the area sort of along Highway 16 in general. And so to determine that, we're going to, and that's going to be going on throughout the project as we tackle some of our questions related to how to keep these predator and parasitoids population healthy. We'll be doing a lot of trapping, and that trap catch that we obtain will allow us to start to catalog what is present, and we'll use what are called morphological keys. So we will look at we will use recognized tools that other taxonomists have put together to help us to determine from the various mostly external body characteristics of the arthropods, what species they are. And we'll also use DNA barcoding, which is something that's been around now for, well, at least a couple of decades. And and in that methodology, we sequence a small portion, about 600 letters of the DNA in a specific region of the DNA that all insects and spiders will have. And if other people have sequenced that same spider species, for instance, and have identified it using morphological keys, we can then look back, we can look at our sequence and compare it to that sequence and say, hey, we have this particular spider species. So it's a nice way of assisting the work of identifying all of these different creatures. And also it allows us to move a little bit faster, because nobody can be an expert in all of these different groups of organisms. So it helps in terms of moving a bit faster. It's you know, the gold standard, of course, is having a real taxonomic expert in a particular group look at something and confirm that that is what it is. But it really helps us to get into the neighborhood of what these things are. And then we hope to develop a real comprehensive catalog of the regional agroecosystem arthropod biodiversity over the course of this 2-year project. And then I would hope as well into the future, I hope this really kicks something off that's a bit bigger and longer term. Activity 2 is also being run by Megan, and this is where she will ask various questions related to how to keep, you know, what causes potentially particular herbivore populations to be prevalent in an area, or even more importantly, what causes particular natural enemies, so predators and parasitoids, what helps their populations to be healthy in an area to help control herbivores. And one of the things that is often discussed in terms of keeping natural enemy populations healthy is what's called SNAP. And so if you have a crop, around the crop might be a forested land or a hedgerow or a grassy area or road with a ditch. And in those areas are plants that are not crop plants, obviously. And those plants and maybe other landscape features might provide shelter for natural enemies to hide out in particular times of the day or the year, or during different times of their life cycle. They might provide nectar or pollen to natural enemies as well. Natural enemies, we hope, are going to go and kill and lay eggs in or eat some of the herbivores, but many natural enemies also rely on other food sources, things like nectar and pollen from plants that are not necessarily crop plants. And then alternative prey as well. There might be times, for instance, in a crop when the herbivore that a producer would be concerned with isn't available for the predator, for instance, but you'd want the predator to stick around. And so if there are different types of prey, different insects feeding on some of the plants that surround the field, that allows the predator to live in that surrounding area and eat those different things and stick around for when it's actually needed in the crop itself. So we want, and beyond that too, this relates then to land management, like what sort of features surround particular fields, and how does that encourage SNAP and encourage natural enemy populations to be healthy. So in a bit of detail there, how can we better manage SNAP to increase natural enemy ecosystem services? So what are good management techniques to encourage a lot of that resource for predators and parasitoids? Also, in much of our region, as you know, I grew up in Calgary and spent a lot of time between Calgary and Edmonton and Calgary and Regina, those areas there where we have large, quarter sections or even full sections or several sections of field crop and not much around it other than roads and maybe some ditches here and there. But the agriculture in this region is often quite different than that, with smaller fields, forests oftentimes coming right up to the edge of fields or near to fields as well. And so this gives a bit of an opportunity to study how land use types and how landscape structures like forest field interfaces, how that influences the abundance of crop pests and natural enemies as well. And there's a lot of good data out there, aerial data and other types of data that allow us to look at shapes and sizes of fields and how much perimeter and forest influence or other influence might be there too, and use those to help us determine what might be good for increasing the prevalence of particular predators or parasitoids. And also looking at the particular predators and parasitoids that we catch in a field, which communities, so which groupings of these different arthropods indicate really good pest control ecosystem services. And Keith has already mentioned many of these sorts of creatures. On the right, you can see Scaphanotis marginatus, which is a carabid beetle, a ground beetle that feeds on slugs, for instance, and snails and other things. This is in the middle is a lycosid spider, a wolf spider with the genus name Pardosa. That's a ground hunting spider that might eat various creatures that are on the ground that are feeding on parts of the plants or sheltering down below and then feeding higher up during the daytime or nighttime. And then there's a little parasitoid wasp there that I picked that I suspect is probably in the region, a family of parasitoid wasps anyhow. And parasitoid wasps will find caterpillars or even eggs of pest insects, and they'll lay their own eggs into those eggs or the caterpillars, and then the babies will hatch and feed on the egg or the caterpillar and kill it that way. Those parasitoids are often very tiny little creatures, just a few millimeters long at most, and so they can be pretty hard to identify morphologically. There are definitely many experts in the world that do that, but it's a lifetime of practice and learning to do that. So using DNA barcoding will allow us to really survey some of these groups. So we plan to focus on carabids and spiders and parasitoid wasps in terms of natural enemies, but other things might come up too. Keith mentioned rove beetles, for instance, little staphylinid beetles. And there's a few others that might pop up as well. Because we're learning what's actually in the region right now. It's going to, some of these ideas that we have may shift as we talk to producers and as we talk to nature by catching things and seeing what we actually find in different fields and field margins and the neighboring forests. We're also planning through MyTax to hire a bachelor's degree student, a summer student, so a summer research assistant who will help with the overall research in the course of 2 summers. But that individual or individuals, depending on if it's 1 student for 2 summers or 2 students over each of 1 summer, we haven't found the person yet. We're working on that. Is going to take on a slightly riskier project, but one that could be pretty cool and interesting. And what we are hoping for them to do is to collect spider webs from in and around fields. And of course, spider webs will have a bunch of little insects that have been caught in them. And then we can do with DNA barcoding, you would take an individual insect and take 1 leg off, sequence its DNA, and then it would be exactly matched to that insect. Over the last few years, there's been a growth in something called environmental DNA analysis. And environmental DNA analysis finds bits of DNA that are floating around in the environment and sequences all of them at once, and then separates out those sequences. And so you can tell mostly everything that was actually in a sample. So oftentimes this has been done in aquatic ecosystems where our lab has done that, in fact, where we sample water and the little aquatic invertebrates shed DNA into the water, and we just collect their DNA and sequence it, and we can tell what was in that. And we've done in our research program as well. We've looked at, for instance, fish gut, the diets that, so fish, trout, for instance, all the food that they eat, once it gets into their stomach, it's pretty mashed up and pretty hard for anybody to identify anything in there. But we can take a little bit of that mashed up food and sequence the DNA out of that and figure out which insects or other creatures the trout was eating. And we are hoping to do this as well with spider webs and a bit of a test case thing that might give us a little bit more of an idea of what these spiders, so web spiders, orb web type spiders, are catching around fields. So to some extent, that's letting the spiders be our consulting biologist and do the catching, set up traps and capturing things for us. But it also will tell us a bit about the food web as well, what spiders are actually catching and eating and what ecosystem services they're providing. So that will be that'll be an interesting little project on the side, but I think that it could be pretty useful long term if we can make it work. It's a bit of a new thing, but it's also, I think, pretty cool. So to conclude this short talk here, what we're looking to do is develop a better understanding of the arthropod pest issues facing farmers in the region. And we're planning on investigating methods of enhancing natural enemy populations through agro-ecosystem habitat management. So we want to determine what makes for really healthy habitats that encourage the natural enemies of the herbivore insects and help to keep crop damage below economic thresholds. And that is my talk, and I'm happy to take questions. Thank you for the opportunity.
So I have to make the proviso. I have to read the arranged statement. Funding for this project has been provided by the Northern Development Initiative Trust and the Canadian Agricultural Partnership. The project was developed by the Climate Change Adaptation Program for Agriculture and was carried out by us at UNBC. Just a note, the funder has had zero input into this. This is entirely kind of separate from outside influence. I'm going to talk about the pollinator project that started early last year. So pollination, as you know, it's an essential ecosystem service. And in most flowering plants, between 75 and 88% of species, depending on who you read, animal pollination is necessary for those plants to reproduce. In this part of the world, that means arthropod pollination, insects for the most part. In other places in the world, things like bats and birds might be super important for pollination, but up here, it's largely arthropods, and of the arthropods, it's mostly insects. If you look at the food production side of things, about 30% of crop production requires animal pollination, but I'm going to show you some evidence in a minute here to kind of suggest that even if you're growing something that doesn't require animal pollination, you probably want to have animal pollinators around. And the fact is that as of right now, there are no cheap, efficient, widely available alternatives to pollination. So it's really important that we know who our pollinators are and that we try and do some managing to keep them around, because no one wants to have to hand pollinate all their squash, right? That's what the photo at the bottom on the left there is, right? I'm sure you may have hand pollinated squash plants in your day. It's not much fun. It's fussy. It takes forever. And, you know, in terms of alternate strategies, I could dress my dog up in a bee suit, but it's not going to make him an effective pollinator. So even if you're growing things that aren't explicitly in need of insect pollinators, there's starting to be some really strong evidence that if you're growing things like canola, that technically, yeah, it's a wind pollinated plant. In cases where you have pollinators around, so you can allow pollinators access to these plants, generally yields go up. Or the quality of the product goes up. So the left is from a study on cotton, and in the two situations where they allowed for pollinators to be present, the open and the cross situations, they had higher fiber weights, so better quality product than when they did not allow cotton plants to be exposed to pollinators, which is in the self mode on the left there. On the right, same thing for canola. If you allow pollinators access to areas where canola is growing, you get higher seed biomass. That's money in your pocket as a farmer, right? As a grower, compared to states when you don't permit pollinators to be around. So it's really important to kind of understand that pollination is this ecosystem service that we really take for granted. Even if a plant is not specifically evolved to require a pollinator, pollinators often can really benefit outcomes when you're growing plants, because they facilitate outcrossing among different plants, right? Because they move pollen around between areas. And the other thing to kind of keep in mind here, and I'm sure everybody's aware of this, but I do like to make the point that honeybees are not the only pollinator. It's quite funny when I tell people I'm working on pollinators, often they think I'm working on domesticated pollinators. They think I'm working on honeybees. But the fact is that, so the figure on the left here with the circles, if you look at those circles, the numbers in them describe the number of plants that can be pollinated by a given group of pollinators. So Apidae, that would be your honeybees, your bumblebees. Halictidae is sweat bees, which are little solitary ground nesting bees. And then the Syrphidae are actually pollinating flies. They're hoverflies or flower flies is the term sometimes you'll hear. And you'll notice the vast majority of plants, at least in this study, they looked at economically important plants. The vast majority of plants that can be pollinated by honeybees can be pollinated by a bunch of other stuff. And all of these other species, they're free. They live in nature. You don't have to feed them. You don't have to look after them. They're just there if you take care of business, right? From the right is an interesting piece of work. It's a meta-analysis that kind of lumped together. They looked at data from multiple studies, and they found that, so if you use the honeybee as kind of the zero line, so the whatever amount of fruit set you get with honeybees as the zero, when you look at quote unquote other bees, so native pollinators that are bees, or pollinating non-bee species, things like hoverflies, you actually get increases in terms of the amount of fruit set you get out of a given crop. And this is really compelling evidence that we need to start thinking about native pollinators as kind of a tool in the arsenal, as something we can take care of, so that we don't have to deal with all the futzing around that involves, is involved in doing kind of domesticated pollinator use. One of the reasons why it's really nice to have a nice diverse pollinator assemblage wherever you're trying to grow plants is that when you have a bunch of different species of pollinator, they all kind of do slightly different things. And the best illustration I've come across of this is from this study by Cusser and colleagues, and it was on cotton. And they looked at how different pollinators interacted with different parts of cotton plants and during different times of the season. And they found that when they blocked off certain parts of the plant from being pollinated, they saw a drop in specific kinds of pollinators that would visit. So they looked at butterflies and flies in addition to traditional kind of pollinating bees. And they found that while the bees showed up a little bit earlier in the season, butterflies and flies were around a little bit later in the season. And that if you looked at where on the plant different pollinators visited, if you lost one of those groups of pollinators, you would get quite a sizable decrease in the amount of product you got out of the plant, because bees don't visit the same flowers on the plant as butterflies and flies do. So the more diverse a community you can get, they suggest you get more of the plant gets pollinated, more flowers get visited generally. But also this applies to things that flower over the course of a season, that have an extended kind of flowering time. If you only have one kind of pollinator around, you're not going to get complete pollination. The other thing that pollinator biodiversity does is it provides resilience in pollination. And so by this, I mean that when you have shifting environmental conditions, so that flowering plants flower at slightly different times every year, right? And in the figure on the right, this is from a study that compared the timing of emergence of 7 different groups of bees relative to the blooming of apple trees in the northeastern US. But this kind of idea applies anywhere. And they found that in years when the apple trees kind of bloomed at the quote unquote normal time, it was mostly andrenid bees that were around to pollinate them. But in years when the apple trees bloomed a little bit later than normal, different groups of pollinators were present to pollinate the trees. So when you have wide diversity in pollinators, you have always the ability to have plants be pollinated because there's always something around that will pollinate it. Whereas if you just depend on one group of pollinators and that is all that's there, well, if things change, if you have some, shifts in things like the timing of flowering, you may end up with pollination failures down the road. So kind of with this as the background idea, we went out last summer and we inventoried pollinators between basically McBride and Telkwa. So BC Central Interior is likely to be a relative winner when it comes to climate change. And the idea is that, for people who grow food, there are some questions about which native pollinators that are already here are compatible with the agriculture that's going on. If you're thinking about growing something new or different, do you have a compatible pollinator here? Well, we don't really know because we don't really know what the native pollinators are. And then kind of the second piece that there are some questions about are how land use and management practices in agriculture affect biodiversity, in particular of pollinators. And so kind of with those as the two knowledge gaps, we went out and we managed to hook 18 sites into the project. We had some wonderful landowners who were just so great, so accommodating about letting some random person show up and put little bowls full of soapy dish soap all over their property. We went out and we sampled it at 18 sites across a 600-kilometer kind of east-west corridor. These represented a variety of production systems, but slightly more than half were kind of forage crop producers, largely alfalfa. Yeah, and so we used a couple of different trapping styles. We used these things called vane traps, which hang on a pole at the side of the field. And essentially what we're doing here is we're using color to attract pollinators. This isn't like, this is a nice general kind of trapping method for pollinators. I know you will have seen for pests sometimes a very specific, like a pheromone bait that gets used that's for one particular pest that you want to monitor. We wanted to use kind of like a general signal to all pollinators to tell them, hey, come and be in the trap. We'd like to know what you are. And so we use color. In the case of vane traps, we use blue vane traps. There are yellow ones on the market, but blue is kind of the color that's recommended for bees in particular. But the second kind of trapping method we used are called pan traps. And so these are little bowls that I painted with some UV reflective paint. I used 3 colors, yellow, blue, and white, which are kind of the traditional colors that are used in pollinator sampling. And these are generally pretty effective for many solitary bees and hoverflies. And so about once every 3 weeks, I would show up at a site and I would put these out in a transect. So I'd put them on the ground like 3-4 meters apart, 24 traps at the edge of a field and then in the interior of a field. So hopefully we could get a sense of how forest edges, so how forest patches on the landscape are influencing what's happening in pollinator communities that are actually available for agriculture and out in fields. So with that, I collected all these samples and I came back to the lab and I started sorting. And we took a fairly broad perspective on this. Many different arthropods were considered as potential pollinators, including crab spiders, that's the little silhouette at the far left, longhorn beetles, moths and butterflies, a subset of flies. So, usually if it was kind of a hairy fly, I figured, yeah, it might be a pollinator. And then bees. And for the most part, we focused on bees that have pollen carrying structures or are otherwise modified to transfer pollen, usually parasitic bees. You can tell pretty quick that it's a parasite. There's a couple of kind of obvious traits. So I didn't consider those as pollinators, but most other bees I considered to be pollinators. And with these samples, I would take out representative kind of types. So morphotype is the term we use. It just means something that looks different. So every new looking thing is considered a new morphotype. And usually that corresponds to a species, but sometimes not. And we took out 665 items that I thought, yeah, these might be pollinators. And we sent them off to Guelph and had them DNA barcoded, which Dezene kind of briefly explained. And once we had species identities for these, I went into the literature and I looked at whether, this species that we, have a DNA signature for it. Is it a pollinator? Is it a beneficial for other reasons? Or is it a pest? Or maybe some combination of those. And it turns out over the course of 24,000 items plus in about 180 samples, there are at least 224 species of likely pollinators, which was really quite amazing. I was really kind of blown away at that. Wide diversity of potential pollinators. We found spiders, we found beetles, flies, bees, butterflies, and moths, all of them pollinators. But in terms of what dominated samples and in terms of what was kind of the most numerous and that you're actually going to see when you're outside looking for pollinators, these are kind of the top 5. So 4 species of bumblebees. I don't use common names because they're far too easy to confuse. But these are kind of the key pollinators that you'll see pretty much on every single type of crop in, across the region geographically. Across the region geographically, and they show kind of like hugely generalist preferences in terms of their ability to interact with different kinds of crops. The 5th species on this list, Lazyglossum leucozonium, is actually quite a small bee. But again, this is the other thing in terms of the number of sites these occur on. They're hugely widespread, and they appear to be pretty happy generalists in terms of their pollinating habits. These are the other 4 of the top 5. So the top 2 left photos there are both of Bombus vancouverensis nearcticus. And this is meant to be a bit of an ad for why DNA barcoding can be so helpful, is because to look at those top 2 left photos, you might think, oh, these must be different species. But the fact is they're not. There's just massive color variation within a given species. The bumblebee on the top right there is Bombus mixtus, sometimes called the fuzzy horned bumblebee, I think. And the way you recognize that one is it's got a little bit of a black stripe across the end of its abdomen and then a bright golden bum. And then the bottom right there, that's Bombus perplexus. So the perplexing bumblebee, and they're typically yellow through the thorax and the top of the abdomen. And then the bottom left is that Lazyglossum bee that's kind of the 5th most common pollinator. This is not to scale. These guys are quite small. They're kind of, they would easily fit on one side of a dime, just to give you a sense of perspective. They're not very big at all. In terms of the other common bumblebees that you're going to see that appear to be quite widely spread, Bombus melanopygus, bottom photo on the left there, it's got the 2 bars of kind of bright orange. And then Bombus terricola is the photo on the right. And it has kind of, it's all black except for a stripe of yellow up at the top of its thorax and then 2 on the abdomen. I haven't included any photos of Bombus rufocinctus because it is a mimic of almost every other bee out there. We found 3 or 4 different color patterns for Bombus rufocinctus. So they're actually quite hard to identify unless you get them under the microscope. In terms of what you can look out for locally, if you're in the western part of the area that we surveyed, so I'm talking like out near Telkwa, there's quite a lot of Bombus oppositus, which I think is the white-shouldered bumblebee is the term. That's the photo on the bottom left. And then also in certain areas, Bombus frigidus, so the frigid bumblebee, the photo on the right, looks exactly like Bombus mixtus, except it has clear yellow at the front of the thorax. I wasn't expecting to see them in agricultural areas. They're physically quite intolerant of hot temperatures, but they did turn up in a few places and typically were kind of clustered around kind of the Fraser Lake area. They seem to turn up the most, but keep your eyes peeled for those. If you're farther to the east, as in kind of McBride and the Robson Valley, you may see Bombus cryptarum, the cryptic bumblebee, which is the right-hand most photo here. And they have quite a remarkable white tail. And I didn't see any of those west of McBride, I think. So they seem to be kind of an Alberta species that hangs out kind of to the eastern end of the range that we looked at. But it's not just about bees. The other key pollinator group that I think really deserves some more attention is the hoverflies. So there's one family within the order Diptera. This one family, we found 63 different species within this family of hoverfly. And we know these are highly adapted to pollinate. If you look at them, you can tell quite a lot of them look an awful lot like bees, right? They're mimics. So they're often yellow and black, or they are shiny and colorful. But there's so many species of them. Most sites actually have somewhere between 10 and 30 different species of hoverfly that will be there providing pollination services over the course of the season. And in terms of what these guys do for you, they're incredible, because quite a lot of them also have larvae that eat aphids. So these hoverflies function as beneficials in addition to being pollinators. They're really quite incredible. And we don't know enough about them here in Canada. Now, in terms of understanding how the landscape is affecting pollinator diversity in areas where you're trying to grow plants, generally, if you look at kind of this is the raw data, and you compare, what I'm calling edge samples, so those pan traps that went right next to the forest, if you compare those traps with the pan traps that were in the interiors of fields, edge samples showed kind of slightly higher abundance, so lots more individual items, but not that many more species. So if you look at kind of the third line down there, the species richness of likely pollinators really is not that different to speak of. However, when you get down in the data and you start actually looking at kind of the group level, so at the family or the genus level, in particular, there's a huge difference between the diversity and abundance of megachilids, which are mason and leafcutter bees. It's quite a bit higher near forests. And mason and leafcutter bees are often the bees that are most important for things like alfalfa. So it's pretty interesting to see that of the 16 species of megachilid that we detected, there are 3 hoplitas, which is the group on the left, that photo on the left, that's a hoplitas. So there's 3 species of hoplitas and 2 species of osmia, which are sometimes called orchard bees. We only ever detected them on the edges of forests, never in field interiors. And so the way to recognize osmia, they're usually fairly small bees. And oftentimes, if you get the light right, they're kind of metallic blue. That's pretty typical for most osmia. Hoplitas are a little harder to identify, but often they will have that kind of long, skinny abdomen. But there's kind of no, unless you get them under a microscope, you can't ever really be sure it's hoplitas. But yeah, so of the 16 species that we found of megachilids, 5 of them only occurring on forest edges, which tells you that forests must be important for this group. The other thing to kind of notice, and this is really a provisional finding, but it's that those hoverflies, so those pollinating flies, they tend to be more abundant near forests. There's a similar number of species that show up on forest edges and in field interiors, but if you look at the count, so the number of individuals, usually the counts from edge traps are about 10% higher. This doesn't necessarily translate to a 10% difference in pollination service, but it's certainly something that's very interesting. And the group within the Surfidae, so the group of hoverflies that showed the greatest difference in abundance between edge and interior samples are the Xylotini, which are these kind of larger bodied, kind of bigger hoverflies. And so I'm still working away on that, but I just thought it was interesting that the larger bodied hoverflies in particular seem to be really affected by forests, and they seem to be much more abundant where there's forests nearby. In terms of the species that we were finding in edge samples and in field interiors, there were a lot more species that were kind of exclusive to edges. So of the kind of 63 total hoverfly species, 14 of them were found only in forest edges. 8 of them were found only in field interiors, and then the rest were found a little bit in both. However, it's not all bad news for field interiors. The fact is that a couple of groups of bees, in particular, everybody's favorite, the bumblebees, tend to be slightly more numerous in field interiors. So not more species, just more individuals would show up in traps in field interiors. The typical field has about 3 different species of bumblebee and about 4 different species of halictids. So halictids are sweat bees. They're typically quite small, but they are active pollinators, and they are highly adapted for that. You can see in these photos, these are all of various halictid species. And you can see in the photo on the left, in particular, those hairy legs that help them to carry pollen. The 3 species of bumblebees, so Bombus appositus, Californicus, and Melanopygus, we did see these more often in field interiors, in comparison to kind of those common bumblebees I talked about a little bit earlier, which were generally found almost everywhere. They didn't seem to show a preference, but these 3 species, appositus, Californicus, and Melanopygus, seem to occur more often in the interiors of fields rather than on the edges. And then in terms of the sweat bees, so the halictids were more numerous on field interiors. But the interesting part here is, yes, there were more sweat bees in field interiors, but they were slightly less diverse. So when you got a sample out of a field interior, usually, yes, there would be a lot of halictids, but they would be 2 or 3 species only. Whereas the samples from field edges, you would see somewhat fewer in terms of numbers, but you would see generally more diversity in the halictids you did get on those field edges. So in terms of the kind of effective haying, which is the one intervention that happened on, I think it was 6 or 7 of the 13 or 14 forage producers where we sampled pollinators last summer. The one intervention that happened on about half of those properties was haying. And so just provisionally, I kind of looked at the numbers of pollinators that occur before and after haying in comparison to places that are growing some kind of, or being used as some kind of like pasture or forage crop, but that weren't hayed. And what was really interesting is that when you look at just hayed fields, so just the blue, oh, I might have gotten this backwards. So if you look at, yeah, I did get this backwards, I think. If you look at hayed fields early in the season, diversity, the overall diversity of pollinators was about 120 species, and it goes down to slightly shy of 100 after haying. But if you look at the early season and late season number of pollinators in sites that were not hayed, it goes from kind of 99 to 100 species over the course of the season. Like it's very similar. So what's interesting is that haying decreases diversity, but more species are present on fields that are going to be hayed early in the season. And I'm not really sure why that is, but I think it's quite interesting. But in terms of, if you break it down and you look at individual groups, you see a very consistent decrease in the number of species of hoverflies, so syrphids, and megachilids, which are those mason and those leaf cutter bees that can be so important for alfalfa production. In terms of what I can tell you right now about how we should be managing for pollinators, I have to say it's pretty limited. I don't have a lot of analyses done yet. We've just finished getting the species identifications done. But generally speaking, if you can keep the landscape complex, so plant lots of different things and keep some forest patches, if you can keep it complex, you're probably going to do pretty well. Anytime you can retain kind of edges and semi-natural habitats, there is a wealth of papers out there. I'm running out of time here, so I'm just going to skip past the quantitative evidence. But at the end of the day, plant and pollinator biodiversity are tightly linked. So if you can make sure there's lots of different kinds of flowers out there, you're going to have lots of different kinds of pollinators. That's the trick here. The figure on the bottom left is pretty obvious, right? The line goes up. There are more bee species when you have more different kinds of flowers. The one on the right shows you that as you convert over land to one single kind of crop, as you increase kind of the homogeneity of the landscape, you see a decline in the richness of the bee species that are out there, no matter what you are growing. It doesn't matter what the crop is. All that matters is the amount of area that you've converted over to doing just one thing. So if you are thinking about pollinator conservation, what's critical here is just to start by preserving what you have. Keep your forest patches, keep your native plants. Encourage floral diversity however you can. So minimizing disturbance to semi-natural habitats is one way to do it. Like, don't just mow it all down, right? The other thing is to perhaps explore some alternative cover crops and or plant some species mixes. So one of my landowners was trying out an oak pea combo, and that seemed to work out pretty well. I've also seen a number of papers recently actually about the value of legumes for pollinators. And if you're thinking about going all out and doing some pollinator enhancement, I could highly recommend the pollinatorpartnership.ca. They have eco-regional planting guides that are specific to every single ecoregion in BC. They're excellent. You can also specifically search for pollinator-friendly plants using their tool, which is the Find Your Roots tool. And then the Xerces Society is a nonprofit from the States for invertebrate conservation, and they have some of the best technical documents and guidelines that anybody puts out. They're excellent. I would highly recommend looking at their materials. Okay, I think that's it.